In 1985, the Soviet research vessel Akademik Fedorov was reportedly running routine surveys in Antarctic waters when the crew hauled up something that made them question marine biology itself. A sea spider, but not like any sea spider anyone had documented. Its legs spanned nearly 30 inches, and it wasn't a freak. The coldest water on Earth keeps doing this. Isopods the size of footballs, jellyfish trailing tentacles 120 feet long, longer than a blue whale, worms that move like animated skeletons. And here is the strange part. Descend into the deep ocean, a completely different world with completely different rules, and you find the same thing happening again. Giant isopods, colossal squids. Two separate cold worlds building giants independently. For over a century, scientists have argued about why. Cold water holds more oxygen. Depth slows life down. Predators vanish. Every theory explains one world and fails the other. The answer is stranger than either side of the debate, because polar gigantism and deep-sea gigantism look identical and run on completely different engines. One powered by oxygen, the other by time. The ocean ran this experiment twice, and both times it built monsters. Colossal, formerly described in 1881, holds the Guinness World Record as the largest sea spider species known to science. Leg span up to 70 centimeters. Nearly 28 inches of leg attached to a body barely 7 centimeters long. Researchers examining large specimens have measured the leg span at roughly 7.9 times the combined trunk and proboscis length. That ratio isn't rounding error. It's the architecture of the animal. The genus Colossal itself is uniquely concentrated in the Southern Ocean. The majority of known species occur there, many found nowhere else. Antarctic isolation has produced a radiating clade of giants concentrated in the coldest, most oxygen-rich shelf water on the planet. Those legs aren't purely structural. The gut of Colossal doesn't stay in the body. It extends into every walking limb, a branching digestive system distributed through the legs themselves. The sex organs follow. Reproductive anatomy housed in the walking limbs, extending outward from a central trunk that is, in terms of tissue mass, almost an afterthought. Woods and colleagues described this in their 2017 work. Relative to other arthropods, pycnogonids have small trunks and long legs, extremely reduced abdomens, and large proboscides. The University of Hawaii Research News expressed it more economically. Sea spiders use their legs as gills and their guts as hearts. Sea spiders have no gills, no lungs. The 2018 study by Lane and colleagues in the Journal of Experimental Biology confirmed what anatomists had long suspected. Pycnogonids rely entirely on cutaneous respiration, oxygen diffusing through pores in the cuticle and absorbed directly from surrounding water. A 2017 Royal Society paper from Woods and colleagues called it transcuticular diffusion, probably via pores. No specialized breathing structure exists. The skin is the respiratory organ. The legs are the majority of the skin. Surface area equals oxygen intake. A larger animal with more leg surface absorbs more oxygen than a smaller one. In a body plan where your integument is your lung, being bigger literally means breathing better, provided the water around you contains enough dissolved oxygen to sustain the demand. That provision is the first engine. Cold seawater at saturation near 0 degrees Celsius carries roughly 10 to 12 milligrams of dissolved oxygen per litre. At 25 degrees Celsius, that drops to 6 to 8 milligrams. That 30 to 40% difference follows directly from Henry's law. Gases dissolve more readily in colder liquids. Cold also suppresses metabolic rate in ectotherms. Lower temperature slows the chemical reactions that consume oxygen.
Both effects compound in polar water. Supply increases, demand decreases, and the ratio of oxygen available to oxygen consumed shifts substantially in the organism's favor. More oxygen in the water, less oxygen needed per gram of body mass. For an animal that breathes through its legs, that ratio governs everything. Cyanea capillata, the lion's mane jellyfish, belongs to cold water environments, primarily in the Arctic and subarctic North Atlantic and North Pacific, rather than the Antarctic. But it demonstrates the polar gigantism pattern at a scale that makes everything else feel modest. It's confirmed as the largest jellyfish species on Earth. The most frequently cited historical specimen was found in Massachusetts Bay in 1870. A bell reported at approximately 7 feet across, roughly 2.1 meters in diameter, with tentacles reportedly stretching over 120 feet. That measurement is a 19th century anecdotal account rather than a peer-reviewed observation, and it should be held accordingly. Modern confirmed records typically describe bells reaching around 1 meter in diameter, but the 1870 report has circulated in zoological literature for 150 years, and if accurate, those tentacles extended beyond the maximum confirmed length of a blue whale, which tops out at around 100 feet. Cold northern waters, dense with plankton, producing a jellyfish with tentacle arrays that dwarf the largest animal alive. Glyptonotus antarcticus was first described from the South Shetland Islands. It lives on the southern ocean seabed from the intertidal zone to more than 600 meters depth, in the family Catiliidae, order Isopoda. Most isopods, the same taxonomic order that includes the woodlice living under garden stones, measure between 5 and 15 millimeters. Glyptonotus reaches a typical maximum of 9 centimeters, with rare individuals documented up to 20 centimeters. Its biology is calibrated for an Antarctic existence, carnivorous and cannibalistic, feeding on echinoderms and whatever it can scavenge. Females brood eggs in a marsupium for more than 18 months, an extended investment that trades reproductive speed for offspring quality in an environment where food arrives unpredictably. A 2005 genetic study suggested Glyptonotus antarcticus may actually represent several distinct cryptic species rather than a single taxon, evidence of the deep in situ radiation that Antarctic isolation tends to generate. 3 separate animal lineages, pycnogonids, cyphozoans, isopods, all reaching outsized dimensions in Antarctic or cold polar water. None are closely related. The pattern crosses phyla. In February 2007, the New Zealand long-lining vessel San Aspiring was targeting Antarctic toothfish in the Ross Sea at approximately 1,500 meters depth. As the longlines came up, the crew found a squid still holding onto one of the fish. It didn't release. It was dying by the time it reached the surface, entangled in the lines, attacked by the toothfish, unable to be returned to the water. Weight when frozen, approximately 495 kilograms. Post-thaw total length, 4.2 meters. Mantle length, 2.5 meters. Sex confirmed as female by dissection showing ovaries. That animal now sits at Te Papa Tongarewa in Wellington, New Zealand. The museum describes it as the most intact adult colossal squid ever caught, and as of 2025, the only complete specimen of Mesonicoteuthis hamiltoni on public display anywhere in the world. The species was first named scientifically in 1925, when G.C. Robson identified the head and arms of a specimen recovered from a sperm whale's stomach. 82 years passed before a reasonably complete adult was preserved and studied. Its eyes, each 25 to 35 centimeters in diameter, are the largest of any known living animal. The beak of the 2007 specimen measured 42.5 millimeters in lower rostral length. Larger beaks have been recovered from sperm whale stomachs, which means larger colossal squid exist, and nobody has collected one. Mesonicoteuthis hamiltoni doesn't occupy 1,500 meters throughout its life. Small juveniles under 40 millimeters mantle length are found in surface waters down to around 500 meters. As they grow, they descend.
Later juveniles are estimated at 2,000 meters. Adults are inferred primarily from where their beaks appear in the stomachs of sperm whales and southern sleeper sharks, which puts them at a minimum of 2,200 meters. The animal sinks progressively into darker, colder, more isolated water as it matures, trading proximity to surface productivity for distance from the predators that concentrate in the productive upper ocean. In 2010, Rui Rosa and Brad Siebel published Slow Pace of Life of the Antarctic Colossal Squid in the Journal of the Marine Biological Association of the United Kingdom, Volume 90, Issue 7, pages 1375 to 1378. Rosa and Siebel built a metabolic model for a hypothetical 500-kilogram Metabolism hamiltoni using data from smaller deep-sea cranchiid relatives, scaled up allometrically and temperature-corrected to 1.5 degrees Celsius. No live adult colossal squid has been studied directly. The numbers are model outputs, not empirical feeding records. The outputs are striking regardless. Estimated mass-specific routine metabolic rate, 0.036 micromoles of oxygen per hour per gram. Projected daily energy consumption, approximately 45.1 kilocalories per day. Translated into prey mass, that figure comes to approximately 30 grams of food daily. 30 grams. For 500 kilograms. The same model implies a single 5-kilogram Antarctic toothfish provides enough energy for the squid to survive up to 200 days. The paper's own abstract drew the ecological conclusion: the colossal squid isn't a voracious predator capable of high-speed pursuit. It's an ambush predator, sit and float, using the rotating hooks on its tentacles to ensnare prey that approaches. Not a hunter, a waiting machine optimized to run on almost nothing. Bathynomus giganteus was formally described in 1879 by A. Milne Edwards. Family Cyrillanidae, order Isopoda. Glyptonotus antarcticus belongs to the family Chaetiliidae. Different families, same order. Both independently evolved large body size in extreme environments separated by tens of millions of years of separate evolutionary history. A 2025 Zoological Science review placed the scale of that divergence in measurable terms. The deep-sea isopod, Bathynomus dodolinei, weighed 34 grams against the common woodlouse, Armadillidium vulgar, at 0.1 grams, a factor of 340 in body mass for animals in the same taxonomic order. Deep-sea giant isopods can survive more than 5 years without feeding, sustained by an enlarged stomach and extremely low basal metabolic rate. When food does arrive, studies have documented that they feed explosively when food is available, consuming a large fraction of their body weight in a single meal. Long fasting, overwhelming feast. The biology is structured entirely around patience. The giant squid, Architeuthis dux, belongs to the family Architeuthidae. The colossal squid, Metabolism hamiltoni, belongs to Cranchidae. The Natural History Museum London is explicit. They aren't closely related and belong to different families of squid. Both sit within the order Oegopsida, but their family lineages diverged in the Cretaceous. Molecular clock estimates place the separation of these major Oegopsid lineages at 60 to 100 million years ago, before the mass extinction that ended the non-avian dinosaurs. The common ancestor of both was a squid, but not a giant. Each lineage independently built an enormous body plan. The physical evidence of that independence is written into their arms. Giant squid tentacle clubs carry serrated, tooth-lined suckers. Colossal squid tentacles carry rotating hooks, a structurally distinct mechanism evolved within Cranchidae for an identical predatory purpose by an entirely separate anatomical route. Two solutions to the problem of seizing prey, derived 60 to 100 million years apart, producing animals that overlap substantially in size and depth range without sharing a large ancestor. Architeuthis dux reaches confirmed maximum lengths around 13 to 14 meters, much of that tentacle. Mesonicoteuthis hamiltoni reaches greater mass. The 2007 Tepapa specimen weighed nearly 500 kilograms at 4.2 meters total length. Longer versus heavier. The same ecological niche,
approached through different body geometries, built by different evolutionary histories. Now the mechanisms, because both sets of giants need explaining, and the two explanations aren't the same. The oxygen temperature hypothesis was formalized in 1999 when Chappell and Peck published Polar Gigantism Dictated by Oxygen Availability in Nature. Analyzing 1,853 benthic amphipod species across environments, they concluded that maximum potential body size is limited by oxygen availability. The framework has governed the field since. Cold water holds more dissolved oxygen, 10 to 12 milligrams per liter near 0 degrees Celsius, against 6 to 8 milligrams at 25 degrees Celsius, following Henry's law. Cold also suppresses metabolic rate in ectotherms, lowering oxygen demand even as supply increases. Both effects compound in polar water. Supply goes up, demand goes down, and the ratio of available oxygen to consumed oxygen shifts in favor of large bodies. For a sea spider breathing through skin pores across 70 centimeters of leg surface, that ratio governs viability. In warm, oxygen-depleted water, a body that size would fail. The surface area would be insufficient for passive diffusion to sustain the mass. In Antarctic surface water, it functions without difficulty. Woods and Moran made the mechanism precise in their 2020 Integrative and Comparative Biology review. The key variable is the ratio of oxygen supply to demand, and cold-driven reduction in metabolic demand is at least as important as slightly higher solubility. The hypothesis has its strongest empirical support in amphipods. Chappell and Peck's original dataset of 1,853 species demonstrated the correlation across environments robustly enough that the 1999 paper remains foundational after 25 years. In sea spiders specifically, the experimental results complicate the picture. In 2009, Woods, Moran and colleagues at the University of Montana published a direct test of the oxygen hypothesis in Antarctic pycnogonids in Proceedings of the Royal Society B, volume 276. The study exposed sea spiders of different sizes to reduced oxygen and measured whether larger individuals declined in performance disproportionately. If oxygen constraint governs body size, animals with worse surface area to volume ratios should struggle more as ambient oxygen falls. The result? No significant interaction between body size and dissolved oxygen on performance. The paper's conclusion was direct. The data don't support the oxygen hypothesis for pycnogonid gigantism, and explanations must be sought in other ecological or evolutionary processes. A second test by Shishido and colleagues in 2019, again in Proceedings of the Royal Society B, examined thermal limits in Antarctic sea spiders and found large individuals no more negatively affected by warming than smaller ones. Two independent experiments, in the flagship taxon for polar gigantism, both came up negative. Verberg and colleagues added a further complication in their 2013 functional ecology paper. Oxygen diffusivity, the rate at which oxygen molecules move through water, decreases as temperature falls. Cold water is more viscous. Despite higher oxygen solubility, cold water may deliver oxygen to a respiratory surface more slowly than warm water delivers it at lower concentrations. Verberg argued that polar gigantism might arise as an adaptation to managing that viscous delivery problem rather than as a consequence of oxygen surplus. Being larger could help overcome the constraint imposed by cold water's physical thickness. Woods and Moran's 2020 review concluded the hypothesis may apply most meaningfully to organisms relying on passive diffusion, eggs, embryos, juveniles, rather than fully grown adults with developed circulatory systems. The honest summary: cold polar water does hold more dissolved oxygen. The pattern holds robustly in amphipods, and the oxygen temperature hypothesis expands the range of viable body sizes in polar environments. But it isn't a clean mechanism governing all polar taxa equally, and the experimental evidence from sea spiders points towards something more complex. Reduced predation in the Southern Ocean, where durophagous shell-crushing predators are largely absent from the Antarctic shelf.
seasonal phytoplankton blooms that provide concentrated energy windows, and physiological factors not yet fully disentangled. Antarctica subsidizes gigantism through multiple overlapping pressures. Oxygen is the most elegant part of the story, not the whole story. The deep ocean is cold, but temperature alone doesn't explain what lives there. Between roughly 200 and 1,000 meters depth, the ocean's oxygen minimum zone develops, where aerobic decomposition consumes dissolved oxygen faster than mixing replenishes it. A 2025 Zoological Science review documented the vertical profile directly. Dissolved oxygen above 220 micromoles per liter at the surface, dropping to approximately 50 micromoles per liter in the oxygen minimum zone at around 1,000 meters, then recovering to 130 to 220 micromoles per liter in deeper water below it. The abyss isn't anoxic. The colossal squid was caught at 1,500 meters in adequately oxygenated water. Oxygen deficiency doesn't drive deep-sea gigantism. Food scarcity does, alongside predation reduction, metabolic suppression, and extreme longevity, all of which interact. Marine snow, particles of organic material drifting from the productive surface layer, degrades as it falls. By the time anything nutritionally useful reaches 1,000 meters, most of the caloric content has been consumed by organisms in the layers above. Prey encounters are rare. Biomass declines with depth below 200 meters, making predator-prey encounters increasingly infrequent. Organisms that expend energy actively searching for food in this environment lose more than they find. Below the mesopelagic zone, where most vertebrate predators concentrate, an animal also faces substantially reduced predation pressure. Not predator-free, sperm whales and sleeper sharks dive deep, and their stomach contents document what lives there. But the selection pressure for fast growth and early reproduction that governs shallow water life is dramatically lower. When metabolic rate is slow enough, you burn very little staying alive. The colossal squid's Rosa and Seibel model demonstrated what that means concretely. 30 grams of prey per day for 500 kilograms of animal. That economy isn't unique to Mesonicoteuthis. Bathynomous giant isopods fast for more than 5 years between meals. The Greenland shark, Somniosus microcephalus, lives between 272 and 392 years. Verified through radiocarbon dating of eye lens nuclei, the longest confirmed lifespan of any known vertebrate. Deep-sea bamboo corals from the Gulf of Alaska, studied through radiocarbon dating and CMAR-CA growth band analysis by Rourke and colleagues in 2005, reach ages of 75 to more than 125 years for individual colonies. The glass sponge Monoraphis tuni has been estimated at approximately 11,000 years old based on growth rate analysis. Extreme longevity is the norm at depth, not the outlier. If growth is slow, because cold and pressure suppress metabolic and growth rates, and you survive long enough, body mass accumulates over timescales that faster metabolizing animals never access.
The deep-sea gigantism literature maintains multiple competing hypotheses. Predation release allowing slow growth to proceed uninterrupted, metabolic suppression under cold and pressure reducing maintenance costs, food scarcity selecting for efficiency and the capacity to fast, and, over evolutionary time, Cope's rule, the documented tendency of animal lineages to evolve toward larger body size when selection for fast reproduction and early maturity weakens. No single mechanism commands consensus. The honest position is that deep-sea gigantism arises from these factors in combination, weighted differently across taxa. What the competing hypotheses share is the central role of time. Whatever the specific route, deep-sea gigantism is an accumulation phenomenon. Bodies built over decades under conditions that don't end them before they're finished. In Antarctica, the environment actively subsidizes size by making oxygen abundant relative to metabolic demand, and by concentrating food during seasonal phytoplankton pulses. In the bathypelagic zone, the environment passively permits size by removing the pressures, predation, starvation, competition, that normally prevent animals from surviving long enough to grow very large. One is a subsidy, the other is a loophole. The most persistent challenge to this two-engine framing invokes Bergmann's rule, the biological principle published by German naturalist Carl Bergmann in 1847. Bergmann observed that within a genus or among closely related species, individuals in colder climates tend to be larger than those in warmer ones. Polar bears are substantially heavier than brown bears. Large northern ungulates outsize their southern relatives. The pattern is real, and the mechanism for endotherms is well understood. A larger body has less surface area relative to its volume, loses heat more slowly to cold air, and maintains core temperature more efficiently. Natural selection in cold climates favors the body plan that retains heat better. For warm-blooded animals, mammals and birds, the empirical support across latitudinal gradients is substantial and documented across hundreds of species comparisons. The polar bear comparison isn't wrong. Polar bears are Bergmann large in a mechanistically supported sense. The rule doesn't transfer to marine invertebrates, and understanding why is the key to the whole distinction. Sea spiders, isopods, jellyfish, and Antarctic shelf invertebrates are ectotherms. Their body temperature tracks the water around them. They generate no internal heat, so they can't lose heat to retain. A giant Antarctic sea spider isn't large because retaining body warmth confers a survival advantage. The animal's internal temperature already matches the surrounding water. There is no heat retention advantage available to exploit. Bergmann's logic has no purchase on the biology of an ectotherm. What governs size in cold water marine ectotherms is a different set of mechanisms. Oxygen availability relative to metabolic demand in polar surface water, predation dynamics, evolutionary history in Antarctic isolation, and in the deep sea, the compounding effects of slow metabolism, scarce food, and extreme longevity. The pattern superficially resembles Bergmann's rule, large animals in cold environments, but it arrives through completely different physics applied to organisms that operate under completely different thermal constraints. Jordan's rule, a Bergmann-like principle for fish noting that cold water species tend to have more vertebrae, doesn't straightforwardly predict body size either. Bergmann's rule itself has documented exceptions in ectotherms. Some reptiles and insects are smaller at high latitudes, a phenomenon documented as converse Bergmann's rule. Even the endotherm version isn't universal.
The second common framing of the question is the sharper one. If the deep sea is cold, why doesn't the same mechanism that produces giant Antarctic sea spiders also produce them in the abyss? It's precisely the right question, because the apparent similarity between these two cold environments does the most work to obscure what's actually different about them. Both are cold. Both produce giants. If temperature were the governing variable, the same mechanism should operate in both, and giant sea spiders should be as common at 3,000 meters as they are on the Antarctic shelf. They're not. Glyptonotus antarcticus doesn't occupy abyssal plains. Messonychoteuthis hamiltoni doesn't compete with shallow-water Antarctic pycnogonids in the same ecological space. The Antarctic shelf at 500 meters is cold, seasonally productive, oxygen-rich, and home to organisms breathing through their skin in a resource-abundant environment. The bathypelagic zone at 1,500 meters is cold, food-scarce, predator-light, and home to organisms that survive by consuming nearly nothing for months at a time. Same temperature range, completely different resource environments, different selection pressures, different giants, different engines. Two squid lineages separated by 60 to 100 million years of independent evolution, reaching similar scales through separate anatomical solutions. The isopod pattern repeats the structure, Glyptonotus in the Southern Ocean, Bathynomus in the deep, in different families. A molecular phylogeny by Ralpak and colleagues found evidence for multiple independent colonizations of the deep sea by separate shallow-water isopod lineages, each generating morphological radiation in the new environment. Multiple independent origins, similar adaptations emerging each time from different starting points. Brandt's 2005 Antarctic Science Review noted the cluster of traits that defines Southern Ocean benthic organisms at scale: gigantism, slow metabolism, longevity, and a reduced number of offspring, combined with late maturation. That same cluster characterizes deep-sea organisms through completely separate lineages. The convergence is consistent enough to reflect physical constraint rather than coincidence. When the range of viable strategies narrows sufficiently, food scarce or oxygen demand suppressed, predation reduced, time extended, the body plans that persist over evolutionary time cluster in a specific region of possibility: large, slow, patient. Multiple lineages enter that space from different starting points and find overlapping solutions, not through coordination, but because only a narrow range of answers satisfies the physics. So the next time someone tells you cold makes things small, send them to Antarctica. Then send them to the abyss. The ocean has run the gigantism experiment twice, once with oxygen, once with time, and both times it built monsters. A sea spider with legs like a skeleton's fan. A jellyfish, reportedly, longer than the largest animal that has ever lived. A squid that eats 30 grams a day and outweighs a bear. Cold doesn't shrink life. Given enough of it, given enough dark, enough depth, enough time, cold is where life finally finds room to become enormous.